It's such a privilege to be speaking to you guys today. In addition to thanking the organizers of the conference, I also want to send a special thank you to Barbara Picower and the folks at the JVP. I'm actually the recipient of a Picower Clinical Fellowship, which has been instrumental in protecting my time and giving me the space to ask these important questions in Professor Li Wei-Sai's lab. I have two goals for you today. The first is I'm excited to share with you the research I'm doing. The second is to convey to you the personal journey that I've taken over the last 4 or 5 years that has led me to this point today where I stand here before you convinced that poverty is a significant disease that is affecting the lives and well-being of children today in America. So what you can see here that I've plotted for you is the number of people living below the federal poverty level in the United States over the last 60 or so years. And one thing that is most striking about this graph, in my opinion, is the increase in the amount of people living under the federal poverty level over the last 45 years. Initially 23 million in 1973, and over the last 45 years, over doubling to 46.7 million people. That translates to about 15% of the population of the United States that's living below the federal poverty level. I think it's important to start with some definitions. What does it mean to be poor living in America today? So in 1963, a group of policymakers and economists got together at the US Census and discussed what does it take to actually make ends meet in America today. And what they decided was that a family would need to make about 3 times the amount of the cost of food at that time to be able to make ends meet. Now, over time, that number and that figure has been adjusted for inflation. But what it translates to today is that a family of 4 living at the poverty level or at the federal poverty level makes about $24,000, and that translates to about $16.60 per person per day. I think it's important to take a moment here and think about what that actually means. I can tell you as I frantically prepared for this talk yesterday, between coffee and takeout meals alone, I spent well more than that. Now, a family of 4 not only has that amount of money to spend on food, but they got to make that stretch over housing, over transportation, over healthcare. And interestingly, all of those things, housing and healthcare especially, the cost of those things have increased faster than the rate of inflation. So today, making and rising above the federal poverty level isn't enough. Families actually, it's estimated, need to make twice the federal poverty level to even make ends meet. So when we look at children at risk of living in these households, we find that 43% of all American children are living in a household where families are struggling to make ends meet. That translates to 32 million children. And America stands alone, not in a good way, but we're last in the developed world when it comes to rates of childhood poverty. You know, I knew these numbers as I trained in medical school and in graduate school, but to me, they didn't really resonate with the America that I knew. I was fortunate enough to grow up in a suburb of New Jersey and trained in Boston for both college and graduate school. And the America that I knew was the Boston that we see in the South End. Brownstones, tree-lined streets, cafes on every corner, organic supermarkets. It wasn't until I became a pediatrician getting to work at Boston Children's Hospital and Boston Medical Center that I saw that there was a parallel America. America that existed in the shadows of the America that I knew where poverty ran rampant. And it was a desire to understand that sort of set me on this journey. So if I can take a moment with you, what I've projected over here is an aerial map of Boston. We're sort of above the Charles River here at MIT today at the Symposium. And these are the 2 hospitals that I trained at for my pediatric residency. Boston Medical Center, depicted in red, which is a safety net hospital for the city of Boston, and Boston Children's Hospital, which is a leading academic institution that over my course of residency had moved towards taking more private insurance and less public insurance. Now, if we look at income, median household income over this map of Boston, 2 things emerge dramatically. First, look at the range of median household incomes that exists in one city. A family can be living next door to another family that is making 10 times more money. The second is how close these neighborhoods are. You can literally cross a street in Boston and go from a neighborhood that is well off to one that is in despair. So if we took 2 respective neighborhoods from the catchment sites of these hospitals. In yellow, I've depicted a neighborhood of Brookline, where most of the children go to Boston Children's, and I cared for them in the ED and on the floor there. And in red, I've depicted a community in Roxbury, where a large percentage of the children go to Boston Medical Center. Let's take a look at how the numbers stack up. In Brookline, the median household income is $106,000. The poverty level and prevalence is well below the national average at 4%, and the life expectancy is a healthy 84 to 87. What about in Roxbury? There, the median household income barely scrapes $14,000. Poverty prevalence is an order of magnitude greater. 40% of all individuals are living below poverty. And this is the fact that shocked me. The life expectancy in the same city has a delta of 25 years. That means a family living a mile apart, on average, can expect to live 25 years shorter than a family living in Brookline. This number shocked me when I first learned it as an intern caring for patients. How could Boston have a life, and people living in Boston have a life expectancy that was lower than that of Haiti and most sub-Saharan African countries? But it was actually what I was seeing on a daily basis, straddling these institutions. And I know a couple of people here work at both these hospitals, and I hope they would agree with me. We had the same medications, the same treatments, the same doctors. I would go to both institutions, and yet the outcomes were so different. And it was really that question of understanding why those outcomes were so different that set me on this journey. Poverty clearly does not work in isolation. We just heard a riveting talk from Dr. Burke-Harris that adverse childhood experiences, trauma, all of these things we know are also occurring in these communities. This is a very convoluted puzzle. In fact, we know that poverty doesn't work in isolation. Here, a study run by the US government found that 34% of all children living below the federal poverty level had been exposed to 2 or more ACEs. But interestingly, in a dose-response fashion, the more wealthy and well-off a family was, the decreased number of ACEs they experienced. Nonetheless, people have wanted to try to understand how are these individual forces, poverty, adversity, toxic stress, contributing to health outcomes. A group of economists at Stanford, led by Raj Chetty, set out to ask this question as well as they could using population-level data. So what they did, taking the US Census information, was plot here on the x-axis household income percentile, and on the y-axis life expectancy. And they controlled for race, ethnicity, location, some of the things that they could using using census-based data. And what they found was shocking. There was a 10-year difference in life expectancy between the poorest and richest women, and a 15-year difference in life expectancy between the poorest and richest men. Naturally, I think the question that follows up is why? What is causing this difference in life expectancy? My PhD was actually in infectious disease. I studied tuberculosis, which is known to be a disease that affects the poor globally. So I thought maybe it's infections. Maybe there are higher rates of infectious diseases in people that are living in poverty. And while we've identified a lot of drivers of illness that are associated with toxic stress, there really wasn't anything, infections, cancer, other causes of morbidity and mortality that came up as being isolatedly associated with poverty. It wasn't until I turned to the space of behavioral neuroscience where I found that people were actually starting to ask the question about how resource deprivation and poverty might affect development and health. So in this study done by economists at Harvard and collaborating with some behavioral scientists, they wanted to ask, is there a cognitive hit that the brain takes as a result of poverty? So what they did was they took individuals from the lowest quintile of the socioeconomic bracket and the highest quintile, and they had them perform an IQ test. Now, before that IQ test, they asked them to ponder a situation in which they would have to spend a little bit of money. And that amount of money could either be a low-cost scenario where they would have to spend $150, or a high-cost scenario where they would have to spend $1,500. And what did they find? They found that only individuals living in the poorest quintile had a conditional drop in their IQ and their cognitive performance and their IQ when asked to ponder about a financially stressful situation. That there is a conditional, measurable hit that poverty has on the ability of the brain to function. So then the question naturally becomes, well, what's going on in the brain of these individuals? And people have also started to look at this. So an interdisciplinary team based out of New York, Harvard, and University of California put hundreds of children across the socioeconomic spectra under an MRI scanner. And they asked, are there any differences that we can find in brain volume or brain function? And shockingly, even when controlling for parental education, they found that key regions in the frontal lobe, the prefrontal lobe that Dr. Burke-Harris was just talking about, the volume and the surface area of those regions was directly correlated to the income of a family. They even followed these children over time and showed that even though children might start off in the same point, the children that grew up in richer families had more brain development than the children that grew up in poor families. So now we've moved from a difference in cognitive function to changes that we can see occurring in the brain, So being trained as a molecular biologist and understanding disease as a physician, the question was, well, what are the pathways that are being disrupted and deranged in the brains of children that are exposed to poverty? And here is where the literature sort of ran dry. Now, I would argue, for reasons that we've been already hearing about today, that it's critically important to understand the mechanism through which poverty is having this effect. First, I think we could all relate to that not all children who are exposed to adversity or poverty are going to have the same outcome. There are some children who are resilient and there are some children that are vulnerable, and we need to be able to understand who is at risk, and we need better diagnostic tools. Once we have those diagnostic tools and we can understand who is at risk, we can also then treat them with a novel repertoire of interventions. And finally, I'm going to avoid any political discussion at this conference. It's an interesting time to talk about politics in America, but I'm still a firm believer that data and science can inform policy decision making. So understanding the science is critical. As I hope I've shown to you, human observational studies and population level data is extremely powerful. It can help to highlight these epidemiological and clinical trends that are occurring in the patients that we care about. But as we know, poverty occurs with toxic stress, it occurs with adversity, it occurs with food shortages. So how do we pick out the cause of one individual stressor and the effects of that stressor? I'm going to argue to you today that we actually need animal studies. Animal studies allow us to pick individual forces like poverty or neglect and study them in isolation and allow us to probe the mechanism. So you might be laughing to yourself now. I certainly sort of question myself, am I actually going to propose an animal model of poverty? Clearly, poverty is a complex social phenomenon. I don't mean to downplay that fact. But at its most basic element, isn't poverty simply the severe deprivation of basic needs? And don't all animals respond to deprivation of needs in a certain way? So I would argue that we can start to model some of the forces that are occurring in complex communities using an animal model. And that was how I stumbled upon this model of resource deprivation called the limited nesting and bedding model that was published about a decade ago. And here what researchers tried to do and did was simply deny a mouse the amount of resources that are needed to provide for a nest and a home for itself. So mice instinctually use nesting and bedding material to make an elaborate nest. And it was found that if researchers deprived a mouse of that environment early in life, even after 7 short days, that there was a significant increase in blood cortisol levels in the serum of those mice, suggesting that there was a perceived stress that was occurring as a result of this resource deprivation. Now, I can take an aside here and sort of give you my personal journey here. It's sort of this study that found me in the office of Professor Li Wei Tsai and proposing this study. And through her mentorship and guidance, we sort of refined this model and thought about how we could make it apply to the human condition that we care about. So we've now altered this model, and I'm going to walk you through what we do. So we have I'm going to first walk you through the normal animals. So normally pregnant mice give birth, and as you can see here, they use a combination of nesting and bedding material to build an elaborate nest for which they can feed and care for their pups. Now, after about 3 weeks, those pups are ready for weaning, and they're weaned to a cage where they similarly have the same amount of nesting and bedding material where they can sort of build their elaborate nests. Now, what we can do is we can remove that material and force moms to raise pups on a bare wire grate where they're not able to build that nest and that home for their pups. And then we can simultaneously wean them to a similarly resource-deprived environment. So the question then becomes, does this have an effect? One of the most striking things that we see that this resource deprivation, and I should add here that we do not deprive the animals of food and water. They still have ready access to food and water, and that's not restricted at all. It's just this resource is needed to make sort of this nest. So when we deprive mice of this environment, what do we see? We find that stressed pups that are grown in that environment at the time of weaning from mom have a significantly decreased weight. Now, in the early life stress field of mice research, and I'm sort of quickly learning this field, weight is often seen as a sort of a marker of perceived stress. So we can say that these mice have taken a hit during this period of weaning. The next question, based on the science that I had been elucidating to you in human studies, is are there behavioral effects of this resource deprivation in mice? So to answer that question, I turned to a battery of behavioral tests. I'm only going to present a few of those to you today. But what you can see here is one test that looks at how anxious a mouse might be. In this test, called the light-dark test, there's 2 compartments that a mouse can freely explore. And we ask, how much time does the mouse spend in the dark versus the light? Mice naturally like to spend time in the dark, and we assume that more anxious mice will spend more time in the dark and less time in the light. When we look at our control animals, our normal animals versus our resource-deprived animals, we find a modest but significant decrease in the amount of time spent in the light. Now, at first, I was a little bit dismayed by this modest decrease, but what I found when I actually looked at the individual animals that were being assayed in this test is that there were a few animals every time that consistently performed much worse than the average. Now, I've aggregated data over about a year of doing this experiment in about 80 mice, and what I hope you would appreciate is that stress affects mice differently. There's almost a line that I can draw between animals that I'm going to call resilient and vulnerable. And so to me, this suggested that maybe it's not two populations that are interesting to study, the normal versus the stressed, but three. A normal controlled condition, a group of animals that have received the stress but are behaviorally indistinguishable from the normal animals that I'll call resilient, and a group of animals that seem to fall apart after resource deprivation that I'll call vulnerable. The more I thought about this data, the more I got excited, because isn't this exactly what we see in our patients? We can see patients respond to the same stress in very different ways. It's not every child that is exposed to poverty or disparity that is going to fail. Many succeed. And this resiliency, I found time and time again examples of this resiliency in the literature. And so in many ways, this divergent effect of stress on a genetically similar population of mice suggests that there's something on top of the genome that is affecting their biology and their behavior. Now you might ask, you're doing this one random test of light, dark, and a mouse spends more time in the dark, and so it's more anxious, but are these mice really anxious? And so to answer that, we took mice from each of those three groups, and we did a second test. Now in this test, called the elevated plus maze, mice are asked to venture 4 arms of a maze that's raised from the ground, in which 2 arms are protected and closed off, and 2 arms are open, where the mouse is at risk of falling off the platform. Now, if I was a mouse, I could tell you that I'm afraid of heights, and I would sort of spend most of my time here in the closed, protected area and spend less time venturing out into the open. What do our mice do? We see that our normal and our stressed but resilient mice venture out into the open arms. But our vulnerable animals, they don't venture out at all. They stay protected behind the closed quadrants of the elevated plus maze, suggesting that these mice are truly, truly anxious. So I hope I've convinced you that we can take a model of resource deprivation in animals. We can find behavioral deficits that arise from that model. And so the questions that remain are, or many questions remain. The thing I love about research is when you do one thing, more questions emerge. So what are the brain regions that are important in sort of regulating this stress vulnerability, and how do we measure what's going on under the hood? So when it comes to anxiety, we know that there are key regions in the human brain that are important for regulating anxiety behaviors, the medial prefrontal cortex and the amygdala. Now, interestingly, a lot of work actually done in this institute by investigators like Kei Tai have mapped out the complicated neural networks that regulate anxiety in the mice, and we find that there are similar regions like the prefrontal cortex and the amygdala in mice that also regulate anxiety. So for the purposes of my research, I've chosen to focus on the medial prefrontal cortex. Now that we know where to look, what do we look at? And here I took advantage of having the huge benefit of being in Professor Liwei's lab, a real pioneer in the field of neuroepigenetics. And we decided to look at the epigenome of these animals. Now, you might remember earlier that I told you that all these mice are genetically similar, so it's not a genetic difference that is explaining why certain mice are vulnerable and others are resilient. Now, I know that there's a wide range of expertise and different experts in the audience, so I do want to take a minute to just get us all on the same page with epigenetics. So if for a moment you could imagine that your genome is a library containing thousands of books, where each book is a gene, you can imagine that that book has 2 states. It can either be open and read, or closed and on the shelf. The genome are the books that you inherit. The epigenome are the changes that result in either that book being open or closed. And the epigenome is often impacted by the environment. So there are many mechanisms through which this takes part. One mechanism that I'm going to focus on today involves histone proteins. So the DNA that you inherit from your mom and dad comes wrapped in these histone proteins. And chemical modifications, sort of caused by the environment, can either cause that DNA to be loosely wrapped around those histone proteins and more open, or tightly wound around those histone proteins and closed. Now, we know that in order to get genes to be expressed and present in cells, we need transcription factors, and those transcription factors require binding to open regions of the DNA to facilitate gene expression, or that open book. However, you can imagine if the DNA was tightly wound around histone protein, and the transcription factor wouldn't be able to bind, and so you would get less gene expression. So we can use this, and we can find and ask in different cells which regions of the genome are open and which regions of the genome are closed. So we took our resilient and our vulnerable animals and did just that. I'm not going to get into the methodology, but we used attack sequencing, which allows us to essentially look in a cell and say, where are all the open regions in the genome? And when we do that, you could imagine that we get a lot of complicated data. There are about 20,000 genes, 1.8 billion base pairs in mice. So we need a way to sort of represent that data in a graphical way that even I can understand. So we turn to principal component analysis. What principal component analysis does is essentially take all of the regions that are open in all of the animals and say how similar or different are they, and then reduces that to 2 dimensions so we can see how do animals cluster together. And when we do that, we see that the resilient animals, here depicted by the individual sort of triangles, and then the larger triangle represents the group average, cluster in the northeast axis of the PCA plot. Where are our vulnerable animals? They were epigenetically distinct from the stress-resilient animals. You can see here that the stress-susceptible and vulnerable animals sort of fall on the left side of the red line that I've depicted, with the group average being the large blue square. This, to me, was incredibly exciting because now we had moved from a behavioral difference. We know that all of these animals have been stressed, but some of them behave like the normal animals and some of them fall apart. And we can simultaneously also distinguish their epigenetic signature in their prefrontal cortex, the region of the brain that regulates that behavior. So then you might ask, well, what are these regions that are open and how are they different? So to do that, we included a normal control, and we can ask, how do the open and closed states in our different stressed animals compare to the normal animals? And when we did this with the resilient versus the normal animal, we found 154 regions across the genome that were different between the resilient and normal animals. And then when we did that with the vulnerable animals, we were quite surprised to see that the vulnerable open and closed regions of the genome were virtually identical to the normal animals. So if I can pause here and build a model for how we're thinking about this, we have normal animals, and we know that the resource deprivation stress, depicted here as the black arrow, can either convert them into vulnerable or resilient animals, as determined by how they perform in anxiety testing. So we know the vulnerable animals develop anxiety, but the resilient animals show no behavior change. However, when we look at the epigenetic changes that are occurring in the prefrontal cortex, we see few epigenetic changes in the vulnerable animals and numerous epigenetic changes in the resilient animals, suggesting that normal behavior in the face of stress requires epigenetic adaptation. So what is that adaptation? Open regions of the genome in the stress-resilient animals, I told you that we're numbering around 154, and we know that transcription factors can bind to these open regions, and we know that those transcription factors recognize known motifs, sequences of DNA that they can bind to and then cause gene expression. So we can ask, in those 154 open regions in our stress-resilient animals, are there any recurring motifs? And in fact, we find one. We found 40% of all of our peaks contain this recurring motif, and it was significantly enriched compared to the vulnerable and the normal animals. And that motif is bound by the retinoic acid receptor. Now, this was really exciting because the retinoic acid receptor already has a known function in nervous system health and functioning. It's known that there are a critical group of neurons important for learning and memory that depend on retinoic acid receptor signaling for the maintenance of function. And if that is withdrawn, if retinoic acid is deprived in the environment, and this has been shown in both human and mouse neurons, that those neurons die, and this process has been implicated in Alzheimer's disease and aging. Additionally, I found a study that used a different paradigm of early life stress and found that maternal separation decreased the level of retinoic acid receptor in the neurons of the hippocampus of mice exposed to that separation, suggesting that maybe it was that increased amount of retinoic acid receptor in our resilient animals that was protecting them from the deleterious effects of that stress. Now, as a clinician, I also got really excited because retinoic acid is a metabolite of vitamin A, retinol. And it's been shown that deficiency of vitamin A causes cognitive impairments. So now I don't want to be prescribing vitamin A. This is still very preliminary, but I do think it is exciting to think, well, if we know that deficiency of a micronutrient causes impairment, could supraphysiological administration of that same vitamin facilitate resiliency in the face of stress? So with that, our future directions, thanks to a lot of the innovative work by a grad student and postdoc in the lab, we're now able to actually look at the RNA, look at the gene expression and how it's changing in these cells. So I hope to correlate that with the open and closed regions of the genome that we're seeing. And then if that's true and the retinoic acid receptor comes up again, I think the next step is to see if vitamin A administration can facilitate and turn vulnerable animals into resilient ones. The retinoic acid receptor story is just one of several ones that we're following up on, so we need to sort of follow up on all the transcription factors that we're identifying. And finally, the spectrum of resilience and vulnerability that I'm talking about is really with respect to one phenotype, anxiety. But there could be many effects that this resource deprivation has, and in fact, we would assume that based on what we know from human data, that poverty has many effects on health and cognition. And I want to bring this back to individuals. There's a large growing amount of epigenetic clinical data, so I think we can use that data that's growing and publicly available to see if these same pathways are being regulated in humans. So I hope today, in the short amount of time we've had together, I've convinced you of a few things. One, that poverty is a significant driver of health outcomes, even in the developed world. Two, we can actually use animal models to dissect how social adversity affects biology, and I would argue it's an important part of the continuum with clinical and epidemiological data and investigation. Two, that early life resource deprivation induces anxiety in a subset of mice, and we call these mice either vulnerable or resilient, and that the resilient animals are able to adapt to the stressful environment and avoid developing anxiety, but this is associated with significant epigenetic changes. With that, I'd like to conclude by thanking Professor Li Wei Tsai. It's been an incredible journey that she supported me on for the last year, and this work is just beginning, so we have a lot more to do. I also collaborate very closely with a talented grad student in the lab, Scarlett Barker, and really the entire lab has been important in facilitating my transition from an infectious disease researcher into a behavioral neuroscientist, and it's been painful for me and them, so I want to thank them for that. I also, as I started, it's a real rarity for a physician scientist to have protected time to do research. And that is exactly what the Jeffrey and Barbara Picower Foundation has done for me, so I'm incredibly grateful to them. I also want to thank various core facilities and computational collaborators at MIT, my fellowship committee at Boston Children's and Harvard in neonatology. Both my mentors are R01 funded basic scientists and neonatologists, so they've paved a way for me to sort of think about how to map this career out. And finally, I feel like I need to disclose to you, I'm actually a vegetarian, and I refused to do mouse work in grad school. But I believe in this project so strongly that I'm willing to compromise that ideal. And all the time I find myself thinking about the families and patients that I cared for in residency, and I think that's important because it bends the arc of this project to making sure that it impacts patient lives sooner rather than later. So thank you, and I'll take questions.
We have time for a couple of questions. If you have any questions for Ravi. Yes?
Have you thought at all, I'm sure you're a little busy,
Yeah.
but have you thought at all about the capacity to model things like housing instability and multiple transitions that are so common among families living in low resource environments?
Yeah. It's such a good question. I think one of the things that has been so humbling to me, learning from people in the lab and people at the Picower, is how closely mouse and animal responses to social stressors are. We in the lab isolate animals and mice, and we find that they become depressed, they gain weight, they stop exploring novel environments. The housing thing we haven't really addressed. I think I was sort of thinking about that with respect to the nest and depriving the resources in the nesting, but it would be interesting to think about animal models that could recapitulate that.
Ravi, this is really amazing research. I was curious whether or not you had thought about testing depriving the pregnancy part and then allowing a normal amount of bedding in the postnatal period. We have found in our research in young kids and families that timing and duration of homelessness or housing instability matters in terms of long-term outcomes?
Yeah, such a good question. I haven't done that personally. I know that there are other researchers, even within the Harvard community, that are actually doing that exact question of sort of depriving in the prenatal period and doing it after. But that would be a logical extension to look at once we validate this as a model.
Great.
Oh, sorry.
I was working with researcher who was working with mice before, and we had we were trying to make some mice, but then the building next door we had all this construction with the noise, we weren't able to do it. So I guess that's also the pressure of having noise and not having the proximity.
Yeah.
Even though they had the same environment, but just the noise that they were looking for the
Yeah.
buildings, it didn't work. We had to enter the buildings that actually
Yeah.
continue our research.
Yeah, I couldn't agree more. For me, it ethically complicates this research a little bit, but I think it makes it all the more important. So.
Thank you, Ravi. So